Yes, Professor Ola Hössjer comes from Stockholm. He is a professor of mathematical statistics and has in recent years acquired both a broad and deep perspective in the field of origins research. Of research in general, but not least in the genetic field. And there he has also been able to apply his specialized knowledge. His talk will be an introduction and overview of the conference theme, which is Review of Recreated Kinds and Baraminology. So go ahead, the stage is yours.
Thank you, thank you. So, thanks a lot, Göran, for the introduction and for the privilege to give this talk. And as Göran said, this will be kind of an overview talk related to the theme of the concept of the conference, a review of created kinds and baraminology. Baraminology, the systematic study of created kinds. So let me start with an outline of my talk. So I will first define the bushes of life or created kinds model, the fact that we read in Genesis that God created plants and animals after their kinds. And then I will give a brief history of the systematic study of created kinds or baraminology. And then there are typically three goals of baraminology. The first goal is to identify the kinds. Which are these kinds which consist of groups of species alive today, or some of them extinct also. The second goal is to find the mechanisms of divergence and speciation within each kind. So we have like a bushes of life model instead of a tree of life. So within each bush we have speciation, and we will hear a lot about that. All the dog animals form a single group, and which are the mechanisms of divergence and speciation within each kind. And then the third goal is, given that we have identified these kinds, the third goal is to find the ancestral history of each kind. And then we will look into humans. Are humans or a kind of its own? So do we have a unique ancestry from Adam and Eve, the first two humans? And then I will wrap up with some conclusions. So let's start with the bushes of life model. So roughly you can think of three models for the diversity of life. The first one is the classical old creationist model, species fixity, according to which God created animals ready, and then they didn't change over the course of history. The second model is also creationist, and the one I would focus on mostly, the bushes of life model, where God created the first generation of each kind, and then later on in the history of Earth, there was divergence and speciation within each kind or within each bush. And then we have the evolutionary tree of life model, according to which all living organisms have a common ancestry from single unicellular organism. And then life starts much further back in time. So instead of 6,000, maybe up to 10,000 years, we speak then of millions and billions of years. So you could say that the bushes of life model is between the species fixity and tree of life model, because it does allow for speciation, but we will see later on that the mechanism of speciation within each bush is very different from the one postulated in the tree of life model. So let's start by giving some examples of kinds bushes. And as I mentioned, this is ongoing research, so that is the first goal of baraminology, to find those kinds and bushes. But humans form its own group. This includes modern humans, but also historical humans like Neanderthals, Denisovans, Homo erectus. They all have a common ancestry with from Adam and Eve, the first two humans. Then great apes, like chimpanzees, gorillas, orangutans, they form a kind or group of its own. And then we have the dog kind with wolves, different kind of foxes. We have breeding dogs and so on belonging to this kind. We have the cat kind, including tigers, lions, leopards, domestic cats, and so on. So these are different examples of created kinds and bushes. But of course, future research will show exactly which are the bushes. But there are good motivations for these being created kinds. So before proceeding further, let us ask the question, is there a biblical motivation for kinds in general? And that leads us to the next part of the talk, giving a bit of biblical motivation for the bushes of life model. We can start with the first two chapters of Genesis. In the first chapter of Genesis, we read that God created plants after their kinds on day 3, and then different kinds of animals after their kinds on days 5 and 6. And finally, humans created uniquely on day 6, and we read, we can find further details about the history of humanity in the next chapter 2 of Genesis. So this is about the existence of the bushes and kinds. But what about the diversification that we in fact could have speciation within each bush? Then we can think of more indirect arguments from the Bible. So animals and humans were commanded in Genesis chapter 1 to multiply and fill the earth. And then an interpretation is that God designed mechanisms for environmental adaptation in the first generation of each kind to make that command of God possible. And then it's very important to also include this limiting factor that at least all land animals, they experience this severe bottleneck with only 2 or 14 individuals per kind surviving due to the flood of Noah that we read about in chapter 6 and 7 of Genesis. So it was like each bush or kind of land animals had to start over again, a kind of extreme bottleneck. And the word Baraminology comes from a Hebrew word, baramin, for created kind, and it was Frank Marsh in the 1940s who coined this term, Baraminology, for the systematic study of created kinds. And as I mentioned when giving you the outline, there are 3 main goals of Baraminology. Firstly, to identify all created kinds. Secondly, to find the mechanisms of divergence and speciation within each created kind. And thirdly, given that we have identified a particular kind, find its ancestral history back to the first generation. And when answering these 3 questions, it's very helpful and important to bring in taxonomy, because there are taxonomic levels of life, and this also has impact when studying the bushes and kinds. So taxonomists, they sort life hierarchically into different levels. At the lowest level, at the bottom, we have species, then we have genus, family, order, class, phyla, kingdom, and domain. And as a creation scientist, you don't believe in that this is a tree of life. So all species, they don't have a common ancestry, but species at the lower levels, they can still have common ancestry, and that leads us to the question, which is a typical taxonomic level of a bush kind? Is it at the species level, genus level, family level, and order level? And already 100 years ago, 2 creationists gave tentative answers to this question. Erik Wassmann, he said around 1910 that it really depends on the studied group, whereas George McCready Price, about 100 years ago, said that often family is a good candidate for defining a particular bush or particular kind. So and both of these predictions turned out to be very good. So if we look at humans, for instance, well, then as I said, all humans have unique ancestry from Adam and Eve, and then it's really humans form a species of its own. Then it's really the species level that is appropriate. But the other three kinds I mentioned, great apes, the dog kind and cat kind, taxonomically they correspond to the family level. But there are many creation scientists that argue that chicken-like birds form a kind of its own, and then it would be defined on the order level. Of course, ongoing research can change this tentative suggestion, but anyhow, the taxonomic level of a kind could depend on the particular kind studied. So let's carry on with a brief history of the Bushes of Life model and alternatives. So up to the 18th century, most Christians were creationists, and even the species fixity model dominated. But even in the young Earth worldview was also mainstream in society. The first edition of Encyclopedia Britannica in the 1770s, it was implicitly understood that the Earth was young, and that there was a long article about the flood of Noah and so on. And we know that, and then Carl Linnaeus, the Swedish scientist who is the founder of taxonomy, he also believed in species fixity, as did most scientists at his day. But late in life, he actually changed his view towards the Bushes of Life model. So that was a brief early example of that. Then in the 18th century, we know that many scientists adopt high ages, in particular after the work of the two Scottish geologists, James Hutton in the late 18th century and Charles Lyell, who popularized James Hutton's work. And this has a big impact on developing different tree of life model. The French scientist Lamarck's evolutionary model, and then Alfred Wallace and Charles Darwin, they published their evolutionary theories about at the same time. And the long ages was a big motivation for them defining the tree of life models, because then they thought that evolutionary mechanisms had time to the diversity all of life if the Earth was old. But even contemporary scientists at their time, there were more and more scholars who not only criticized the tree of life model, but they favored different versions of the Bushes of Life model. For instance, the British botanist William Herbert in the 1820 and the geologist Richard Owen in the 1850s. But in spite of this, when Charles Darwin published his famous Origin of Species book in 1859, he equated creationism with species fixity. And for a long time, among many scholars, it was sort of believed that creationism was the same thing as species fixity. And then when we move on to the 20th century, the tree of life model gained more and more adherents, in particular when the modern synthesis took place in the 1940s. And that's like marrying evolutionary theory with genetics, finding genetic mechanisms, natural selection acting on mutations, and that's also called neo-Darwinism. And that was scientists who were important in this development were the geologist George Simpson and the biologists Julian Huxley, Ernst Mayr, and Theodosius Dobzhansky. And at the same time, in the early 20th century, there were increasingly many creation scientists who favored different versions of the Bushes of Life model. And here we have some of these names, including Erik Wackman and George McCready Price that I mentioned about earlier. And then, as I mentioned, in the 1940s, Frank Marsh coined Baraminology as the systematic study of created kinds. And I would say that from the 1950s, species fixity became a minority position among creationists in favor of the Bushes of Life model, because the data seem to point in that way. So after this brief historical overview, let's now return to the three main goals of Baraminology. The first goal is to identify all bushes or created kinds. The second goal is to find the mechanism of divergence and speciation within each created kind. And the third goal is to find the ancestral history of each created kind once it has been identified. So let's now talk about these three goals. Starting with the first goal, identify created kinds. Then there are three main methods for doing so that complement each other. And they are hybridization or observations, experiments, the so-called cognitive method, and statistical baraminology. And I will explain these concepts in more detail. The first method for identifying created kinds is hybridization. So suppose two organisms or individuals can hybridize and have offspring, since reproduction is an extremely complicated process from the first fertilized cell and then through embryological development. The two organisms, they have to be related in order to have viable offspring. And for this reason, Frank Marsh in the 1940s, he suggested hybridization as a sufficient condition, not a necessary condition, a sufficient condition for a pair to belong to the same created kinds. In order for them to belong to the same species, you require more. Then the pair also need to be able to have fertile offspring. So, but how do we more exactly define hybridization within kinds? Well, the offspring need not be fertile. So donkeys and horses, they belong to the same kind, but mules, their offspring are still not fertile. But it's still an hybridization, proving that monkeys and horses belong to the same kind. And in fact, the offspring need not even survive embryological development, but it has to pass a certain stage of this development. And that was suggested by Siegfried Scherer in the early 1990s as a criterion for hybridization. And then there is a lot of hybridization data, a book by Gray from the early 1970s, and a very comprehensive article by Jean Leiter from 2012. So let's look at a particular example, hybridization within the cat family. And the cat family has 40 species and 14 genera, the next taxonomic level. And here is hybridization data from 10 species and 6 genera, notated G1 to G6. And the crosses in the boxes, they correspond to successful hybridization, and it's most likely incomplete. But you see that there are successful crossings over genera borders. So the data suggests, based on the hybridization criterion, that the cat family is indeed a created kind. But then we can use the other two methods for identifying kinds as a complement, and that leads us to the second method, the cognitive method. And the idea is that it is possible for us humans to use our senses and sort species into groups. However, such a group could include several kinds, because based on our senses, we could conclude that all mammals have things in common. But of course, mammals consist of several kinds. But we can also use our senses to conclude that all foxes have visual resemblance, but it's still only a part of the dog kind. So for this reason, the cognitive method should be used as a complement to other methods of identifying kinds. And then we could also have a sort of indirect biblical motivation for the cognitive method, since we read in Genesis chapter 2 that Adam, he was able to name all the animals. So, and that implicitly must imply, or it tends to imply, that he was able to identify the kinds. And probably the same thing was true when Noah was able to get the land animals of each kind into the ark, that he was able to recognize them using his senses. So let's now apply the cognitive method of the cat family, for which we looked at hybridization data. So we see that there is strong visual resemblance between the species of the cat family. So we can use a cognitive method to conclude that they form a group, and that supports the conclusion that we had already from the hybridizations, that the cat family is indeed a group or created kind. So that was the second method of identifying created kinds. The third one is called statistical baraminology, and it's used for very large data sets, either molecular ones or morphological ones, that is with the forms of different parts of bodies. Very large data sets which cannot be analyzed by human senses alone. You need computers and algorithms to do the grouping. And you often talk about three major steps of statistical baraminology grouping. First, you have a certain list of species, and for each, and then you also have a list of characters. They could be genetic characters or morphological characters, the form of the skull or something else, and then you, for each character, you put you have a black dot if that character is present and a white dot if it's absent. So in this very simple example, we have 4 species and 10 characters. And then in the second step, you compute, for each pair of species, you compute a distance between them based on the fraction of characters that differ. And that gives you a table, in this case with 4 rows and 4 columns, because we have 4 species. We see that species 1 and 2 are very close. It's only character 6 that differs. So the difference is 10%. Only 1 out of 10 characters differ. So we put a 10% in that box, the pairwise distance between species 1 and 2. Species 1 and 3, they differ in 9 out of 10 characters. It's only character 6 that is the same. So therefore the distance is 0.9, 90%. And then in the third step, then we cluster the species into group. And that's usually presented as heat maps, so that small distances corresponds to light boxes and large distances correspond to dark boxes. Small or large distances between species. And then species that belong to the same group, they correspond to these white, larger boxes. So in this case, we have clustered the 4 species into 2 groups, species 1 and 2, because they are very close, small distances, and the species 3 and 4 into a second group, because there are also large distances between the species of several groups. Let me give an example of a heat map of real data. Here we have 34 animal groups or species, and we don't have 10 characters, but 20,000 protein characters. So it's a huge amount of data, and I will actually return to this heat map later when we look at human uniqueness. Then let's now turn to the second goal of baraminology, finding the mechanisms of divergence and speciation within kinds. And as I mentioned early, that the biblical motivation for not only the existence of bushes, but also the speciation that occurred, is that God created each kind to rapidly adjust to change environmental condition. And this includes 2 main features. First, God created diversity in the first generation of each kind or bush. And then, secondly, there were designed mechanisms of change so that we could have very quick changes and adaptation to environmental changes over the history of time. So in this case, we have an example of a bush with presently 4 species at the top. And we have the branching events of the tree correspond to speciation events. So in this case, we have 3 speciation events, and these are due to these mechanisms of change. But and this implies that the observed degree of speciation in each created kind is possible within a young Earth creationist timescale, like 6,000 years, because these design mechanisms of change are very quick and rapid, and it's not generating novel things, but the information they are based on, they were already present in the first generation. But it's also important to point out that in parallel, there are also evolutionary non-design mechanisms of change that are an effect of the fall of man, and they are much less efficient, and they are mostly deleterious and mean loss of information, devolution. Typically, they don't lead to speciation. Maybe they can do that in few cases, but they rather threaten survival of species. And let us now give some examples of created diversity. And there are many versions of it: created heterozygosity, variation inducing genetic elements, epigenetic structures, redundancy, and engineering principles of environmental tracking. So we will hear a talk by Peter Borger on variation inducing genetic elements today, and tomorrow by Tommy Alto on epigenetics. So we will hear more about these different versions of created diversity. Let me just mention a little bit about a few of them. Created heterozygosity, this we will hear more about today when Nigel Crompton speaks about Mendelian speciation. It's really, if we look now at the human group, founded by Adam and Eve, then Adam had 22 pairs of non-sex chromosomes and Eve. So there are 2 copies of his non-sex chromosome, and God created them slightly differently. That's called created heterozygosity. So for each, that means that at certain positions, the base pairs are slightly different of the 2 copies of that particular chromosome. So for instance, Adam's chromosome 1, here we could have, in the coding strand of 1 copy of chromosome 1, we could have a C, and at the other copy, a T. That's a so-called single nucleotide polymorphism, places along the chromosomes where there were differences. And then mechanisms of change could act on this created diversity later on in life. And then variation-inducing genetic elements means that we will hear Peter Borger, he's an expert on this. He will basically, parts of the genome can move from one place to another. So here we see that the lilac part moves from one position of the genome to the other. And the possibility for those elements acting was part of the first generation. And then we will hear Tommy Alto tomorrow talk more about epigenetic structures. This is based on so-called histone modifications. We will hear more about that then. Peter Borger also points out redundancy, and this is also related to Boris Mitgal's talk, that some plants, they have 2 systems for photosynthesis, so they can turn on one and turn off the other. But both were present, the possibility of both systems were present in the first generation, a type of redundancy. And then Randy Gugliuzza has introduced the principle of engineering and environmental tracking. So parts of the cell functions a bit like sonar and radar systems. And then we have a number of designed mechanisms of change, and some of them are these evolutionary detrimental changes. So I will not go through this in detail. We will hear some of this in the talks that come, and also the last issue of Genesis magazine, number 3, the September issue, it goes through many of these mechanisms of change. I will just mention their name: genetic drift and founder effect. We will hear Nigel Crompton talk more about this. Mutations, they could be non-designed or designed. We have crossovers, recombinations. We have natural selection. Peter Borger talking about variation-inducing genetic elements. Tommy Alto about epigenetic mechanisms. And we also have certain plasticity during embryological development that could also lead to speciation within kinds. So we see that, for instance, epigenetic mechanisms, the prerequisite of it was part of the first generation. So it's part of created diversity, but then we also have the mechanism that itself acting over the course of time. So then it's also a mechanism of change. For that reason, both Weiges and epigenetics appears both among created diversity and as a mechanism of change. Let's now turn to the third goal of Baraminology, to actually finding the ancestral history of each kind. So suppose we have identified a certain kind, the bush kind, the dog kind or the cat kind, and then we want to find its ancestral history. And that ancestral history could include several aspects. We have, for instance, biogeography, for instance, environmental changes forcing the organism of a certain kind to move around the Earth. For instance, plate tectonics and continental drift after the flood of Noah leads to species moving to different places of the world. We also have the debris rafting models where animals could raft on debris over big portions of water. We also have climate variations like the Ice Age occurring a couple of hundred years after the flood of Noah that impacted where species could live and also their geographic history. But it's not only the geographic history, it's also the genetic history. With modern method now, we can actually build genetic trees of different kinds once we have identified them. And for two sex species with males and females, we can build male and female trees. For two sex species, the males have Y chromosomes, so we can build a male tree from the Y chromosomes so that more closely related males have a closer ancestry. So in this is an unrooted tree. The tips of the tree are men alive today, and somewhere in this unrooted tree we have the root. And then we could build female trees from mitochondria. Mitochondria are the power stations of the cell, and it has its own DNA. And males and females have mitochondrial DNA, but it's inherited from females. So for that reason, we get female trees from mitochondrial DNA. And it's the same thing. The tips correspond to women alive today, and somewhere along this unrooted tree, we have the root. And Nathaniel Jensen and co-workers, they found in very interesting work that these trees, for a number of different kinds, were, when positioning and timing the root of the tree, that was consistent with the young Earth creationist timescale between 10 and 6,000 years ago. Let's look now more closely into the human trees, the human male Y chromosome tree and the human female mitochondrial DNA tree. So as I said, the tips of the male tree to the left and the female tree to the right are all men or women alive today. And then we have some key questions. Data gives an unrooted tree. Where should we put the root, the most recent common ancestor of all the tips of each tree? And another key question is, how fast do the branches of the tree coalesce? And that is related to when we have found the most recent common ancestor, when did the most recent common ancestor live? And that leads us to the question of the age of the tree's most recent common ancestor. And that very much depends on the concept of mutations. And this is a kind of detrimental mechanism of change that I mentioned about. We had two types of mechanisms of change, the ones that God designed, and mutations, many of them are copying errors when fathers transmit Y chromosomes to their sons, or mothers transmit mitochondrial DNA to their daughters. Sometimes there are copying errors, and that means that branches in the Y tree here, an example with 5 men, the men that are more closely related, their branches differ by fewer mutations. So the numbers here reflect the number of mutations along each branch. So the 2 men to the left here, there are 23 plus 28 mutations between those two particular Y chromosomes. And then we can ask the question, where should we put the root of this unrooted tree? It should somewhere be put so that all branch in the middle of this unrooted tree. And there are methods for doing this. So suppose we put the root here along the longest branch. So 25 mutations to the left of this root and 61 to the right. Then we can turn this unrooted tree to a rooted tree. So the root is the most recent common ancestor of all these 5 males. And then we see the number of mutations along each branch. And now, in order to determine the age of the root, the mutation rate is crucial. And that is the number of mutations per generation. The faster the mutation rate is, the more recently the most recent common ancestor lived. And this is a very important fact now. It's only recently that mutation rates it has been possible to estimate them accurately using the so-called pedigree method. And the pedigree method basically means that in the case of Y chromosomes and male trees, you basically compare the Y chromosomes of fathers and sons and look at the average number of differences when you look at several father-son pairs. It's only recently that the DNA sequencing technology has developed, so you could apply this method to estimate mutation rates, and it has been found that it is a much higher than previously believed, so that the most recent common ancestor lived much more recently than previously believed. And it's of the order of 5 to 10,000 years for a variety of different species. And as I said, you can also apply this to kinds, but it has not been done so much yet, but it has to be done for humans. That leads us to the next part of the talk. I will argue that humans are unique, it's its own created kind, and I will do that in 3 different ways. Firstly, we'll look at a statistical baraminology study to argue that humans are genetically very different from great apes and from other species. Secondly, we will use non-sex chromosomes data to argue that humans' genetic variation is consistent with ancestry from a first couple. And then thirdly, we will go back to these concepts of female trees from mitochondrial DNA and male trees from Y chromosome data to build humans' female and male ancestral trees. So let's start with statistical baraminology, and this is based on a very interesting study by Leitner and Serati from 2019, and this was the heat map I mentioned about earlier. The data consists of 3 species or groups. We have some cat animals, some mice and rat animals, monkeys and apes, 3 groups of humans, modern humans, Neanderthals and Denisovans. We have chicken-like birds and so on. And as I mentioned earlier, there are 20,000 proteins as characters. So for each species, it is marked whether a particular protein is present or not. And then this is the heat map that is a result of this study, and it clearly proves that, or strongly indicates that humans are unique. Because here we have the 3 rows that correspond to humans. It is its own light box, which is very different from the 4 species that comprise great apes. And in fact, in this study, great apes, in terms of whether proteins are present or not, they are more similar to other species than to humans. And I think this is an amazing study because it's based on 20,000 proteins. It's a large data set. And then we have non-sex chromosome data supporting human uniqueness. And then we go back now to the concept of created heterozygosity that we will hear more from Nigel Crompton about. So for humans, the total length of the 22 human non-sex chromosomes is about 3 million nucleotides. And a so-called biallelic SNP, I mentioned about it earlier, is a position along some of the chromosome where there are two possible nucleotides, A, C, G, and T. It could be A and C, that the two possible nucleotides at that particular position. Or in this case, we have a base pair CG or TA. So along the coding strand, that gives a C and a T. That's a biallelic SNP. There are two possible alleles at this position. In this case, a C and a T. And then for each SNP, we can determine the minor allele frequency. That is the frequency of the less common of the two alleles. So suppose this particular SNP that I mentioned, where we have a C or a T along the coding strand, suppose 40% of all humans have a C in that position, 60% have a T, then the minor allele frequency is 40% for that particular SNP. And since there are about 10 million such biallelic SNPs, with a minor allele frequency of at least 10%, we can do statistics of these minor allele frequencies, and that is a so-called minor allele frequency spectrum, and that is a kind of histogram over the minor allele frequency of all 10 million SNPs. And the interesting thing is that the shape of the allele frequency spectrum, it conveys information about the history of humanity. So typically, so an old population typically have a less flat allele frequency spectrum than a young population. And from a creationist point of view, an allele with a large minor allele frequency spectrum typically corresponds to created heterozygosity in the first generation with Adam and Eve, whereas a small minor allele frequency, that is a more recent mutation that has not spread so much in the population yet. Whereas from the evolutionary tree of life point of view, there is no such thing as created heterozygosity. You only have mutations as a mechanism of creating new diversity. So then a large minor allele frequency corresponds to an old mutation and a small minor allele frequency to a recent mutation. And I've written about this myself with Ann Gauger. We published a paper 5 years ago, but also John Sanford and his collaborators, they fitted allele frequency spectra to real data and found that the unique origin model with Adam and Eve had at least as good a fit as the evolutionary models, saying that yes, it is possible with a first couple. So that was arguing for the existence of a first couple based on non-sex chromosome data. But now when we go to male and female trees, we get even much stronger arguments for the creationist model. And let's start with the mitochondrial trees. And I will mention a bit about the history. The first estimates of the age of the most recent common ancestor of the female mitochondrial tree, they were based on evolutionary assumptions using so-called phylogenetic mutation rates. So basically comparing mitochondria between humans and chimps and assuming that they had a common ancestry 6 million years ago. And based on this, the mutation rate was calculated. And I said the mutation rate determines when to put the age of the most recent common ancestor. And based on that, the age of the most recent common ancestry was determined to be about 150,000 years ago, in line with the so-called out of Africa model. But then in the mid-1990s, the first estimates of the more reliable pedigree method came along. I illustrated earlier the pedigree method for Y chromosome. It's similar for mitochondrial DNA. The mitochondrial DNA is circular, but it's the same principle. You compare mitochondrial DNA between mothers and daughters, and you count the fraction of differences for many mother-daughter pairs. And the first estimates of the mutation rates based on this more reliable pedigree method, they gave conflicting results, but generally the age of the most recent common ancestor was found to be at least an order of magnitude larger it should be. It says smaller here. It was found to be at least a order of magnitude larger. And then in Nathaniel Jensen, he pooled all these studies and he obtained a most recent, it's correct. The mutation rate was found to be much higher, and then the age of the most recent common ancestor was at least one order of magnitude smaller. But then Nathaniel Jensen, he strengthened these arguments. He pooled all these studies and using publicly available real data from secular scientists, and he obtained a most recent common ancestor less than 10,000 years ago, consistent with Eve, the most recent common ancestor of all females alive today. And he also identified 3 major branches going out from the root, because and corresponded to the 3 daughters-in-law of Noah, because all women alive today, they can trace their female ancestry from one of the 3 daughters-in-law of Noah. So it really also confirmed that. Then we go to the male tree, that the Y chromosome human tree is also consistent with a young Earth creationist timescale in terms of human history. And the historical development is quite similar. In the 1990s, the first estimates of the age of the most recent common ancestors several 100,000 years ago, they were based on low quality DNA data, because it was very difficult to sequence Y chromosomes because of lots of repetition in the DNA sequence using DNA sequencing methods. But in 2019, Nathaniel Jensen and Holland, they pooled several studies with high quality DNA in order to get better estimates of the mutation rate using the pedigree method. And they found that the most recent common ancestor lived about 4,500 years ago. And that is consistent with Noah, because all males, according to a biblical creation model, all males can fit, are descendants of Noah, because only Noah, his wife, and his 3 sons and their wives survived the flood. And the sons, they are descendants of Noah. So all males alive today can trace their ancestry to Noah, and that goes well along with the age of the most recent common ancestor he found. And 2 years ago, he published his book Traced, where he not only identified Noah as the root of the tree 4,500 years ago, he also identified 3 branches going out from this root, and that biblically corresponds to the 3 sons of Noah. And he also identified branches further down along the tree, and hypothesized or found strong evidence for that they correspond to different persons living a few generations after Noah that we read about in Genesis chapter 10. But not only that, using the Y tree with a young Earth creationist timescale, he was also able to confirm more recent historical findings, like the American African slave trades during the 17th and 18th centuries and so on. So that leads me to wrapping up my talk, giving a summary and some conclusions. So the study of created kinds, it involves 3 main goals. Firstly, to find all created kinds. Secondly, to find mechanism of divergence and speciation within each kind. And thirdly, given that we have identified a particular kind, find its ancestral history, for instance, geographically or genetically, using male or female trees. And this is an intensive field of research with lots of new molecular data, genome-wide data, ancient DNA, and many new tools that require lots of computer power and algorithms. I mentioned about some of them. And the results show that data clearly group species into kinds, but still a lot of work remains, because not all species have been sequenced yet. And there are design mechanisms to allow for quick adaptation within kinds that explain the emergence of the bush. And we will hear talks about that later on today and tomorrow. And analysis of mitochondrial and Y chromosome DNA indicates recent ancestry of each kind in agreement with the young Earth timescales. And finally, the result that humans are clearly unique. The genetic analysis that I mentioned about, they confirm the Bible and human history. And a prediction for the future is that the research that will come based on more data, we have large international DNA sequences projects like the Earth Biogenome Project. They will continue to strengthen the Bushes of Life model and human uniqueness. And hopefully more people will realize then how reliable the Bible is and give their lives to Jesus. And let me give a quote from Romans chapter 1, verses 19 and 20. What may be known about God is plain to them because God has made it plain to them. For since the creation of the world, God's invisible qualities, his eternal power and divine nature have been clearly seen, being understood from what has been made, so that people are without excuse. And part of this, God's invisible qualities being seen, that applies to the diversity of life, that we can see such a structure of created kinds and bushes, and that is part of leading people ultimately to realizing that the Bible is reliable and lead people to Christ. So, thanks for the attention.
Now we will have a short Q&A session, just for some 5 minutes or something. Then we will have, later after Peter Borger's speech, we will have a little longer break with some coffee and such things. And you who are speakers, who bring with you literature, you may later then put it on the bookshelves or on the tables over there where we are looking at literature. So, but if you have any questions, Ola, you may just now. Ola, you can take.
Yes.
I want to remember to repeat the question, Ola, so we get this out.
Yeah, a bit of historical interest here. The term baraminology was coined by 1940s or something, if I remember it correctly there. But I just heard this phrase recently. How come we haven't heard of baraminology as a scientific term or something like that previously? Are there any specific reasons why we are just now being flooded with this expression?
Yeah. Thanks a lot for this good question. The question was that the term baraminology was phrased in the 1940s, but it's only more recently that we have heard about this topic, and it's sort of part of the debate. I think one answer is that generally creation science has been marginalized, but still there are other concepts within the creation evolution debate that have been going on. So another possible answer is that the arguments for the Bushes of Life model have been strengthened so much recently. It's the last 10-20 years that we have found various mechanisms of change that really explains that we can have speciation within each bush very quickly. And these are very different from the supposed mechanism of the Tree of Life model that don't produce anything new. They rather erode information. These are mechanisms that are based on information that was already present in the first generation. So they are extremely strong argument for the biblical model, and we will hear more Nigel Crompton and Peter Borger and Tommy Alti. They will also include this in their talk. Also Boris Mitchell will look at the photosynthesis. So I think that's the main reason, I would say, that recent research has really strengthened the Bushes of Life model and hence put back the term baraminology into the center of the debate. Yes?
Yeah, thank you for your presentation. I think that
argument that has strengthened this thing is the orphan genes, which define the families pretty nicely.
Yeah.
That is a very strong argument.
Oh, yes, that's really true, because orphan genes is an example of... Yeah, so the question was that one very strong argument that Pekka Reinikainen say from for the Bushes of Life model is the fact that we have orphan genes, or genes that are unique to specific kinds of bushes, and that is an example of distinguishing features. So if we look for, I mentioned about statistical baraminology, and then you had like 20,000 proteins. You looked at lots of data, but you can look at single features that still can separate some kinds, and orphan genes is one example. We also have, like Peter Borge mentions, about certain types of genes that are like you have Hox genes and things like that. That are also distinguishing features between different baramins. So you can also either look at large data sets to separate groups, or you can look at a singular type of distinguishing feature like orphan genes.
There was a slide, and I don't know if it's possible to go back to it, when you were describing the use of environmental conditions.
Yeah. Was it biogeography or?
Probably.
Was it this one?
Environmental tracking.
Yeah, here. Was it this one? Earlier?
This one here. Well, you wrote that they've got create these kinds rapidly just to change the environmental conditions. Although I think that this is not an unreasonable statement, I think that much more can be said about this, and there is a tendency to think of this then as being a justification then for selection.
Yeah.
Which is not necessary.
Oh, yeah. The comment here by Nigel Crompton was that change of environmental condition can be sort of overstated, so that the question was that change of environmental condition hasn't it been overstated, so that it's always it's interpreted as a selection mechanism, and I agree that maybe in some cases it can be, but in other cases not. So it's really something that as you will give in your talk, you will mention about genetic drift and such mechanisms that don't include natural selection, but you still have a lot of speciation. So it's really So it's not really natural selection is only one of many possible mechanisms of change, and it seems that its importance has been overstated often.